So, hello everyone. I am going to present you a topic on atomic force microscope. These are the contents which will be I going to cover in the upcoming slides. These are the introduction, fundamentals of AFM, and working of AFM, that how AFM works. Some components of AFM, what are the important components of AFM, modes of operations, the how in what modes the atomic force microscope performs, sample preparations, that how to prepare sample for the atomic force microscope, applications where are the atomic force microscope are used, challenges and limitations in the of the AFM technology. This is the introduction of the atomic force microscope. So I will telling you that atomic force microscope was invented by the Gerd Binning. He was the IBM scientist in 1986, for which he got the Nobel Prize. This atomic force microscope comes under the category of scanning probe microscope. So, scanning probe microscope was the microscope which make the 3D images of the at the microscopic level. These are the microscope which helps to make the revolutionize the nanotechnology sector. So, atomic force microscope is a type of scanning probe microscope that enables high resolution imaging surface at the atomic or nanometer scales by physically interaction with the samples. Like optical microscopy, AFM is not limited by the light diffraction, allowing the detailed 3D mapping of the various materials. AFM operates by detecting atomic forces between sharp tip on a cantilever and the sample surface, which is adjusted to maintain consistent force and provide a topographic image with a different modes like contact tapping and non-contact tapping. AFM can study a range of samples, making it invaluable in fields like material science and biology and nanotechnology. So, these are the fundamentals of atomic force microscope, like high resolution imaging, material properties analysis, surface topography, versatile applications like a wide range of applications including material science and nanotechnology. So surface technology, you can say that the surface for the atomic force microscope is very essential for the surface mapping topography. Because by analyzing the surface topography, we can generate different 3D images by which we can see that how is the there the certain movements are there in the surface is are there. So these are the components of atomic force microscope: cantilever, tip, piezoelectric tube, laser, photodetector, computer system. So I will show you for where the cantilever. So say this is the cantilever. My arrow is there. This is a cantilever which is made of the very. It is very sensitive, and in which the tip is there. That is a very pointed tip. So tip is if we can compare the tip like the pen nib. The pen nib is made of 1,000 of atoms, but at the same time the tip of this cantilever is made of only 7 and 8 atoms. So the when it is made of 7 and 8 atoms, so the interaction between the atoms and atoms is used to detect the sample that and to detect the topography and determine the nature of the sample. So this is the very essential that the tip, how fine is the tip, is very essential. Piezoelectric tube is also plays a very crucial role, that it moves in x and y z direction to so that the tip the contact between the tip and the sample is there. The laser is fall on the cantilever, and it see the cantilever and goes to the photodetector detects these photons and sent to the controllers, and then it is given to the computer system and the data is recorded. And so the computer system is used generally for the giving the signals to the controller, and which controller gives signal to the piezoelectric tube, so that the movement can take place according to the signals, and computer also collect the data. So now the working of the atomic force microscope. So these are the here in this figure we can see that the sample is present, the piezoelectric tube is there, the cantilever with the tip is there, the laser is coming and reflecting from, and then again reflecting to from the mirror and going to the photo detector, and then the signal is sent to the controller and computer. So what happens, uh, thus when the sample is present on the is kept on the piezoelectric tube. So when the computer is set, the set point is set, and now the tip is start moving over the sample. So by the interaction of the atomics between the two atoms, the piezo movement of the piezoelectric the movement of this cantilever take place, and that movement is recorded with the help of laser, and these laser get deflected, and the movement of the laser from the center is seen, is captured, and every time when the laser is moved away from the center, it is been recorded, and then again the signal is sent to the controller, and controller set the such way that the piezoelectric tube is move in such way direction that this laser again come to the center. So by this it every time this happens, and the signal is sent to the computer, and the computer made the different images. So these are the like probe cantilever setup. AFM has sharp probe mounted on the end of the flexible cantilever. This tip is extremely small, often with a radius of just few nanometers, enabling high resolution. Approaching the sample, the AFM tip is brought close to the sample surface using the precise mechanism called piezoelectric scanner, which allows for tiny control movements. When the tip is near the surface, it starts feel forces sampled due to the atomic interaction. See, here is the written atomic interaction. So the movement of the cantilever takes place due to the atomic interaction. The forces and deflection. As the tip moves over the surface, it experiences the various forces, generally van der Waals forces and electrostatic forces. These forces cause the cantilever to bend or deflect. The amount of direction of this deflection indicates the presence of the surface. Laser detection system. So the laser is maintained at the back of the cantilever. When the cantilever deflects, the laser beam is reflected onto the position sensitive photodiode. The moment of the laser is spot on the photodiode corresponds to the tip's vertically and horizontal displacement. So the feedback loop. So as already I have told you that how the feedback loop works. The AFM has a feedback system that maintains a constant force between the tip and the sample. When the tip encounters the bump or valley, the feedback mechanism adjusts the tip height and prevent the excessive deflection. This helps to maintain the steady scanning and produce the consistent data. So this happens when the set point is there given into the controller. So, and generally the set point is what? The bring back this deflected laser again to the center. Scanning and data collection. The tip scans the surface and grid pattern, and AFM records the vertical adjustment required to the maintain constant force. This data is processed to the build an accurate high resolution map and sample surface over the topography. So this is the how the image formation take place. So this yellow thing is the image formation, and these are the graph. See, this is a 2D graph. Actually we can see the distance between the two hills, like the topography is given. So now we are seeing the two things, one is 3D and one is 2D. The 3D is seeing that the how is the surface is there, how is surface is looking, and how much is the distance between the two valleys or two peaks is there. So this is shown in the graph. So now the modes of operations of the AFM. So these are the three modes of operation of AFM. This is contact mode, tapping mode, and non-contact mode. So by the figure you can see that the contact mode means the tip and the surface is always being in a contact. So according to this, the deflection occurs when the tip moves over the surface. The tip moves up and the laser get deflected, and when the tips moves down, then again the laser get deflected, so that there should be the continuous contact between the tip can take place. This is the contact mode, and generally it is used for the metal surface, for the topography of the metal surface, or you can say that the hard surfaces, because the tip is very pointed. So if we use for the soft surfaces, maybe chances that the soft surface get destroyed. So the tapping mode is generally used for the it is in the present the cantilever is in the vibration mode. So it taps like just like a stitching machine. It taps one point, and the force is noted, because when the tappings occur, it is occurring at the constant force. But when the material changes, so what happens, the force which the cantilever is acting is now changes, and that force is recorded. So generally the tapping mode is used for the determining the nature of material. What is the nature of material? And non-contact mode is generally used for the biological samples, in which the tip is just vibrating over the surface, very light, and when the bumps occurs, then the atomic force, the attraction force occurs, and due to which the vibration between the cantilever is become very small vibrations, means the vibration get stable. So again the recorded it is being recorded, and when the valley is there, then the vibration very it is the vibration occurs very fast, then again it is being recorded. So these vibrations are recorded such that the topograph we can generate the topography image on the display. So how the samples prepared in the atomic force microscope. So generally the sample to preparation of sample is done very sensitively, because generally the sample preparation take place in the vacuum. When the sample is prepared, it is placed into the vacuum, because no impurities should be there in the sample, otherwise it will occur, it will causes the can't is the cause the tip to the tip get destroyed, or may you can say that the sample get also be destroyed. So it is generally done in the vacuum. To prepare the sample effectively, it may involve the pre-treating steps such as drying, grinding, or filtering, which helps to separate the analyte from the contaminants. Sample condition may need adjustments like pH of solvent composition to improve stability and compatibility. In some cases, derivation is needed to enhance the volatility or detectability, especially for gases chromatography. Additionally, methods vary in specific requirements. For instance, gas chromatography needs volatile, thermally stable samples, while mass spectrometry requires minimal matrix effects to prevent the interface. Finally, the quality control is crucial to verify the accuracy and reliability of the presentation process. The proper sample preparation tailored to each analytical method ensures high quality reliable results. So this is the slide which represents the how what are the things which we do with the help of atomic force microscope. This is a topography images, like we can see that how the topography can be seen, the ups and downs, the curves are there on the surface, how the curve is looking. Force spectroscopy. Spectroscopy is generally that what is the material of the certain substance, what is the nature of certain substance. It is generally with the help of tapping mode. Topography imaging is generally done with the for the you can say that generally for the hard metals, for the strong metallic, with the help of contact mode. And electrical properties are done can be checked by the help of the non contact mode. So these are the three things which can be helpful by using the AFM. So what are the applications of AFM? So there are a wide range of applications of AFM, but here are the few. Like material science. Material science, it is very useful in the material sciences. Structures, properties, defects in the material at a nanoscale level. As already I have told you that this microscope bring the revolution in the nanoscale technology. So this is very generally useful for the nanotech areas. So in nanotechnology, AFM is used in the design of fabrication and characterization of nanomaterials and nano devices. Biology and medicines, it is used to study structure and function of biological molecules, cell and tissues. Semiconductor industries, AFM is used in the manufacturing of integrated circuits and other semiconductor devices. In the upcoming slides, I will show you how the semiconductor industry use this AFM. The image is showing one image I have, which shows the circuit diagram of the semiconductor. So what are the challenges in the AFM technology? So despite of many advantages, FM has also limitations, like the tip is very pointed, so there's a chances of wear and tear of the tip very easily. While scanning other, especially with a hard and abrasive sample, the tip get easily distracted. This can affect the quality of the image and accuracy of the measurement. Another challenge is the size of the sample. See the what happens, the AFM takes very long time for the analyzing the sample. So if we take a very huge amount of sample, then it takes very much time, and this causes the it is also not economic. So generally we take very small size of sample, so that it does not take very much time. So we can only and only can we analyze very small amount of sample, not a very big samples. So the upcoming slides, we have the some video that how atomic force microscope in a contact mode works. So this is the probe. You can see it is moving in the continuously, it is contact with the surface and moving, and this is the graph which is generated. This is a cantilever deflection, and this is a topography image which is coming in the on the computer screen. And it one by one, it covers whole area. So it shows that how the topography get generated, and this is the final topographical image we get on the computer display. So this is in the upcoming slides. This is the image of by the AFM taken. This is the image of nuclear pore. You can see that this is a black. This is a nuclear pore, which is recorded by the AFM, which is captured by the AFM. So what I have telling you earlier, that I will be showing you image that how it is using semiconductor devices. Like, see the topography of reverse engineering a silicon chip structure. This is done with the only help of AFM. So this is so the how the structure is there in the average in the chip. So it is generally done with the help of contact mode. So structure of butystyrene diene, the material properties, so it is done with the help of tapping mode. Collagen fiber is also done with the tapping mode. And fixed bed Benman cell petri dish, it is done with the non-contact mode. So these are the some references where you can take more informations, like nanosurf, which are giving you information about how AFM works, and even they are selling the AFM technologies. And these are the YouTube videos which we can get some more informations about this. So thank you very much.